they can replicate in the plant, but they cannot produce disease because all the genes, all the ORFs are not expressing. So, using this kind of things, we first confirm that whether it is released out from in the plant, and then we put a GFP in that, and then we, after confirmation, we agro-infiltrate them and confirm again that whether GFP portion is intact inside the plant, and then we observe through UV radiation, we observe that the GFP is expressing in the localized tissue where we have infiltrated. So, that indicates that we can use the viruses as a carrier, as a vector to deliver any protein, any gRNA expression construct, any other things. So, for inhibiting the viral infection, the direct targeting of the viral genome and targeting of the host gene, both the things we have initiated in our lab. So, as I am telling you that DNA and RNA virus both are having single stranded, double stranded. In case of RNA, the single stranded RNA viruses are again divided into two types, positive sense single stranded RNA and negative sense single stranded RNA. So, these are the broad range classification of the plant viruses. So, among these viruses, the first target for the genome editing was initiated with the single stranded DNA viruses. Now, we may ask question that why single stranded DNA, because CRISPR-Cas9 works in a double stranded DNA break, but we are using it in single stranded virus, that I will come. But why single stranded DNA virus? Because single stranded DNA viruses, the size of the genome is very small. So, manipulating that genome is very easy, and they are the most important group of plant viruses. Okay. So, they belong to the family Rna Virus. They are circular single stranded DNA molecule encapsulated into a twinned icosahedral particle. So, the virus looks like this twin particle. It is only one particle, but it looks like two particles attached together. So, this family Rna Virus is divided based on the number of particle, vector, host and number of genes into 9 different genera. So, these are the different genus of the virus, and under each genus there are several species of the virus. Under each species there are several isolates and strains of the viruses. Okay. So, among these all these genera, bigomovirus is the most important in all the virus, and they have total 388 species at present, and they are the largest genera containing largest number of species among all the viruses, whether it is human, plant, animal, anything. The bigomovirus genus containing largest number of species, because this virus can be subjected to huge amount of mutation and recombination, and due to that mutation and recombination very quickly new virus species evolved. Okay? And that is why tackling this virus is also very difficult. So, this bigomoviruses causes very important diseases. The diseases can be classified, which is caused by these bigomoviruses, in two different major symptoms. One group is leaf curl disease. The plant showing, you can see this is the tomato plant, how pathetic condition. You cannot even imagine that tomato leaf is there. So, tomato leaf curl, chili leaf curl, cotton leaf curl. So, these are the very much important diseases worldwide, these leaf curl diseases. And then second group is yellow mosaic or yellow vein mosaic, like Vendi, yellow vein mosaic, yellow mosaic of pulse crop, then yellow mosaic of cucurbits. So, this kind of diseases are caused by bigomoviruses. They are transmitted by a small insect, just like our human, dengue and other things, they are transmitted by mosquitoes. The plant viruses are transmitted also by different insects. So, one of such insect is whitefly. So, these are the small white insect. In the zoom you can see this is the insect which feed on the leaf, and they infect from the infected plant, they take the virus and infect to the healthy plant to transmit the disease. Their genome, the bighorn virus genome, there are two types of bighorn virus again. One is called monopartite bighorn virus, which is present mostly in the old world countries. Old world means North America, rest of the part. American countries are called new world countries. So, in the old world countries, majority of the bighorn viruses are of this type, that only single DNA molecule, and this is associated with some satellite molecules which is called alpha satellite or beta satellite. And these are the bipartite bighorn virus, which is mainly present in new world countries, but also present in old world countries. So, they have the two different genomic components, the DNA A and DNA B. DNA A is mainly responsible for replication and encapsulation of the virus, and DNA B is responsible for movement of the virus. So, this is the in nutshell the genome of this bighorn virus. The management strategy for this virus which is available is the insecticide treatment against the whitefly, which cause enormous environment problem. Utilization of the host genes like TY genes in tomato, but in many of the crops we do not have any idea about the resistance gene. Then RNA transgenic technology, which is giving very much promising result in management of the bighorn virus, and latest addition is this CRISPR-Cas9 based genome editing. So, in nutshell, this is the overview of the bighorn virus as such. Now, the question is that why people target a single stranded DNA virus. Now, if you see the replication event of this virus, the virus enters into the nucleus directly. So, when the virus enters inside the nucleus, what happens? The single stranded genome of the virus, we do not know what is doing. Definitely it is taking some primer kind of sequence from the host, that is still we do not know what are the RNA which is supplying as a primer for these viruses, and they using the host DNA polymers, they cause a double stranded molecule. So, always when you see the virus in the plant, you will get a double stranded DNA molecule, not as a single stranded DNA. So, then this double stranded DNA molecule undergoing transcription come out and produce the proteins, those proteins come inside the nucleus again, and then they start replication of the virus. So, it is caused by a rolling circle replication model. So, from this double stranded things again a single stranded molecule produced. So, always for this replication event it has to come through this double stranded molecule, then only the single stranded molecule will be produced. So, if any way we can inhibit this double strand molecule, then the formation of single stranded molecule will not be possible. Okay? So, that is why people try the attacking the virus in the double stranded form using the CRISPR-Cas9 technology. How we can achieve that thing? So, if in a plant if we are expressing the Cas9 and gRNA as a transgenic overexpressing condition, then the Cas9 and protein and gRNA they will produce this complex molecule, the Cas9 gRNA molecule. So, when the viral genome come and start replication and produce this double stranded molecule, they will go and bind and then they can cut, and then the formation of this single stranded molecule will not be possible. So, keeping this point, actually people started to apply this genome editing technology to the viral genome. It is just like you just now compare with the bacteriophage. The same thing happening actually in the bacteriophage. When the bacteriophage comes infect the bacterial cell, the bacterial cell is already having a transgenic CRISPR-Cas9 content. So, they will produce that thing. So, just that thing is replicated in the plant system to target the DNA viruses of the plant. So, these are the examples already people have worked out. So, last one is our paper which we have published very recently in October this year. So, before that there were 6 papers, actually 3 papers came in 2019. So, when I started working there were only these 4 papers. Everyone is targeting different genes of this virus, and in all these cases mainly the Cas9 protein is overexpressed in the plant, and the gRNA is delivered as a transgenic condition or through virus based delivery, the gRNA is given. And in all this, before my work, all this experiment, they showed the application of one gRNA at a time to inhibit the virus, and we have shown that application of multiple gRNA at a time to inhibit the virus. So, now coming into the RNA virus editing, after the discovery of a different Cas protein which is called Cas13a, or a variant of Cas9 which is called FN-Cas9, it has been shown that these proteins, instead of cutting DNA, they can also cut single stranded RNA. So, when these proteins were discovered, immediately people jumped onto the aspect that we can target the RNA virus editing also, because remember that in case of plant, though bigomoviruses are the most economically important viruses, but the maximum number of viruses are single stranded RNA positive sense virus. Okay. So, if we can target the RNA viruses, then it will be very nice. So, what is that? That RNA viruses we can directly deliver. When it will come, RNA viral genome will be replicated, but if in the plant cell, if we have the CRISPR RNA and the Cas13 protein, then they will produce this kind of active CRISPR-Cas9 machinery, and they can bind with the viral RNA and it can be chewed up. Now, in case of DNA viruses and the RNA viruses, the difference is that in case of Cas9, we require a PAM motif for targeting the viral genome, but in case of Cas13, we do not require a PAM motif. So, that is a very important thing, that anywhere, wherever we want, we can target. We do not need any PAM motif, but the problem is that the off target effect is very high, because we have to choose the RNA very critically, because in other cases in DNA viruses, if there is off target also, if the PAM motif is not matching, there will be no cutting. But in this case, if some sequence is matching, 12 nucleotide seed sequence is matching, there may be off target effect. So, we have to choose very carefully the initial spacer sequence identification, then only this technology is very much useful. And the second important thing difference is that in case of DNA virus editing, after double stranded DNA break, the host replication repair mechanism, they try to join those cut ends, as happened to the host chromosome also, and they create mutation, and those mutation many times virus can overcome. Virus can, even with those mutations, because virus is highly mutagenic, highly recombinational virus. So, it can overcome those small mutations, even deletions and additions, and they can replicate. But in case of RNA virus editing, there is no repair mechanism for RNA repair mechanism in the host. So, if the virus will get cut, they will get chewed up. So, there is nothing to be done. So, directly we will get 100% resistance. So, there are several again works already have initiated, published, different DNA RNA viruses people have worked out, and different genes they have targeted to inhibit the virus infection. Now, coming into the now in this aspect, actually we have one project on this inhibition of the RNA viruses. We have a project on inhibition of papaya ring spot virus, which is a positive sense RNA virus infecting papaya. So, we are trying to target the virus in both way, directly targeting the viral genome and targeting some host factor which is responsible for the establishment of the infection. So, this is the aspect that CRISPR-Cas9 against RNA viruses by targeting the host genome. So, different plants have been shown, and the people target one important protein which is called EIF4E. This is a host factor for RNA virus translation. EIF4E is a elongation factor for any protein translation. So, viral genome, when they incorporate it into the plant system, what happens? They replicate. Before replication, they need to produce the proteins. So, that protein synthesis machinery virus do not have, they completely depend upon host. So, if the protein synthesis of the virus, the replicase protein, the coat protein, the movement protein, those proteins are not synthesized in the plant, then plant virus cannot replicate, cannot able to establish the infection. Okay. So, and this is the gene EIF4E or EIF ISO4E. So, these are the genes people have shown that these different alleles of the EIF4E complex, they helps the virus to establish the infection. And how they do? So, this is a virus, RNA virus, which is replicating by this module using this system. So, this virus has at the 5 prime end, the viral RNA is attached to a protein. So, it is not only a RNA molecule, it is also attached to a protein which is called viral genome linked protein. So, this VPG protein, they interact with this host translational machinery, which is having a different protein complex. So, this VPG protein interact with EIF4E and bring the viral RNA with the host translational machinery, and then only the translation of the viral genome takes place. And it has been observed that this EIF4E and VPG interaction is very specific, and it occurs in 3 different domains of the viral genome, the VPG protein, and 3 domains in the EIF4E. So, this is the host component, this is the viral component. So, they interact only in 3 different domains, and those nucleotides are exactly known. So, people have shown that if we do a little bit modification in those motifs, the protein will be exactly produced, they can help the translation of host RNA and other things, there will be no problem. But the interaction of the virus with the RNA translational machinery will not take place. Okay. So, it will not directly knocking out of the protein, it is only doing something where the virus and that EIF4E interaction takes place. That motifs only we have to do editing, and if you can do editing on that, then the virus will not able to recognize the host translational machinery, and the virus replication will be inhibited. So, this is the main aspect. So, mutation or knocking out has to be done, and in this aspect my collaborator Dr. M.K. Reddy and Dr. Manomohan from IIHR Bangalore had made some success. So, they developed a CRISPR-Cas9 construct utilizing the first they identified several EIF4E gene sequence from the papaya, and then they were cloned and identified the guide RNA. 711 base pair guide RNA was developed, not only one target, multiple target, and then they already did the transformation, callus generation and the planglet formation, and they observed the mutation taking place. So, there are heteroduplex assay. So, this is the original virus, wild virus, this is the wild host, means the original EIF4E, this is the mutated EIF4E, and they observed at least some of the planglet there is only one nucleotide mutation, and that mutation is sufficient to change the interaction property. So, we are still we are waiting for the challenge inoculation and seeing those things. So, this is just initial phase of our project. So, we have initiated this kind of work. Now, this is about the overview of those things. Now, I would like to come to the specific topic of our work. I would like to give you the case study with the chili leaf curl virus, which we have done the multiplexed editing of a bigovirus genome, restrict escape mutant formation and disease development. So, before going into the details, first I would like to tell you what we have learned from the earlier works. So, we observed that this technology was applied in 3 bigoviruses, 2 mastiviruses, 1 topicoviruses, all are the single stranded DNA virus. And in all this case, only 1 gRNA inoculation at one time in one plant, and they have shown that best is the target is the origin of replication. If you target the virus at the origin of replication, that will give you maximum benefit. But simultaneously, there are 2 papers come in 2017 and 2019, they give rise a concern. The concern is that many times these DNA viruses evade CRISPR-Cas9 due to escape mutant formation after non-homologous end joining. So, after even non-homologous end joining, whatever mutation is created, the virus can withstand those mutations and can happily replicate. So, it gives rise a major concern for creating a more mutant robust virus. So, it is a kind of drug resistant bacteria similarly like that. So, it is a possibility they have shown in their paper that could be a detrimental effect of genome editing if you want to do, then virus can come out as a more devastating virus. So, when there was a concern, when I went to US for this genome editing training program, my mentor told me that what you would like to do. So, I told sir what people have done I would like to do because 6 months only. So, I would like to take a gRNA from a virus, I will do the genome editing. So, he just staring at me and told then why I will provide you fund, because if you already people have done and you will do that thing in your virus. So, I told sir actually in India we are doing like that, we are studying the literature for what, that what people have done we will do the same thing in our system. This is in general all the PhD students are doing, that why we are studying literature to copy that thing for our system. But in US this is not the system, you have to study the literature to know where is the gap, where is the concern and try to address, so that there must be some novelty. If there is no novelty, there is no need of research. Okay? So, he told me clearly that you have to tell me where is the novelty, what are the things you would like to do, where is the novelty there. So, I read again and again those papers and try to find out that what is the concern, where is the gap. So, I observed that the gap is that escape mutant formation is a question. And why escape mutant formation? Because everybody is trying one position of the viral genome, and if that position is get rejoined, then there is a possibility of formation of the escape mutant. So, I thought a hypothesis that if we simultaneously target 2 or more sites of the viral genome through multiplexed gRNA, then it could reduce this escape mutant formation, because simultaneously if you target many sites, then the viral genome will get chewed up, and again rearrangement of all the things together to form a virus like molecule is very rare chance. Okay. So, with that hypothesis I thought I should use the experimental system. Host is Nicotiana benthamina, which is a model host for plant virology, and the virus is chili leaf curl virus, because already we have in our lab, we have extensively characterized the virus, we have an infectious clone of the virus, and it is only a single molecule of the virus. So, it is easy to handle, because we have to prove this concept of multiplex editing. People have shown in the plant, animal, everywhere, but in the viruses also, because virus is not a chromosome. So, whether we can in a circular molecule, whether we can do the same thing and whether we can get rid of the virus. So, this is our main aim. So, the chili leaf curl virus, I would like to give a little information. This is the condition in the field, this is the leaf curl disease, no chili, nothing. So, farmers are committed suicide in Maharashtra and Madhya Pradesh. The first report was in India during 1930, but after long back we could able to find out there is a bigom virus which is actually causing the disease. The people earlier thought it is a mite, it is something like that, but ultimately we established from our lab that it is a bigom virus which is causing the disease. And during the course of investigation we observed that it is not only one bigom virus, there are 9 different bigom virus which is causing the similar kind of symptom in different location in India. So, it is not only one virus. So, it is very difficult, it is challenging to target all the virus simultaneously. So, we thought that we will first use the chili leaf curl virus, because almost all place of India this virus is definitely present. Okay. So, we thought that we will target the chili leaf curl virus. So, the virus got very much immediate attention during 2014, 15 and 16, because 2014, 15 in Madhya Pradesh there was a huge epidemic. So, you can see the field, nothing, no crop is survived, no yield at all. So, it was a major crop in Khargon, Dhar and Badauni district in Madhya Pradesh, and it was sporadic during 2013, 14, but more than 90% during 14, 15. And farmers' participation is that the leaf curl was less importance 5 years ago, it emerged due to introduction of new high yielding cultivars. That was the farmers' point of view. But we observe that farmers are not getting at all a single money out of this huge acres of land. In 2016 in Maharashtra, you can see this is the condition, no chili production, only the plants and all are infected with leaf curl, and farmers are started reshowing some other crops, so that from that same field they can get some remuneration. The chili, within the chili crop they planted other crops. So it is a huge devastating, it came in the newspaper, and 5,000 acres land during June to March in Jagatmal district of Maharashtra, they observed that this. So, they abandoned the crop. So, this is the condition of chili leaf curl infection in farmers field. So, the virus is belong to this Gemini virus, Bigoma virus under Gemini viridi. This is the particle morphology, twin icosahedral particle. The genome is circular single stranded DNA molecule and transmitted by whitefly. So, this is the genome of the virus. Just like other Bigoma viruses, it also have all these important genes, and along with that, some cases it is associated with a beta satellite molecule. When this beta satellite is present, the severity of the virus is much more. But we try to target this DNA A, because without DNA A, DNA beta satellite cannot replicate. So, if we target the DNA A, we can tackle both the things. So, we developed an infectious clone of this virus earlier in our lab. So, by this process which I have told you, that taking the two origin of replication and genome, it is a dimeric construct, and when we inoculate this construct into the plant, either benthamiana or chili, we observe the leaf curl symptom. So, our virus is in our hand, whenever we need we can produce the disease. So, that is why we took this virus as our experimental system. So, the entire process already I have told that how you will be doing whatever practical you are actually doing, all these steps we have done for means editing the virus. The first step is identification of the spacer sequence in chili leaf curl genome, then in vitro efficacy analysis, then development of the multiplex construct, in-plant activity of those construct, and then assay for genome editing. So, first is identification of the spacer sequence. Earlier also in the practical I told you that for the identification of the spacer sequence, initially we got a problem that we do not have any CRISPR direct or such kind of website available for the viral genome. So, other cases what you have to do, you take a gene, give it into the website will tell you what are the important, could be the important spacer sequence to target that gene. Okay. But if you give this kind of sequence, what they will do, from this sequence, what are the sequence which can target the plant genome, they will tell. But we need the opposite thing, we need a sequence which should not target the plant genome. So, manually we have to do that thing. So, initially we thought that which portion of the viral genome we should take. So, we thought that one definitely region is the intergenic region. People already have showed that this is an important region where if you do editing you can get maximum benefit. Then we choose another region in this area, that is V2 V1 overlap. So, this is the movement protein gene, this is the coat protein gene, as well as this protein also acts as a silencing suppressor. So, silencing suppressor means the host, whenever any virus enters, host try to inhibit the virus infection through RNAi mechanism, and virus try to inhibit that silencing mechanism by some proteins. So, it is just analogous to the elicitor molecule of the fungus. So, fungus initially infect due to the PAMP triggered immunity, they can be excluded, but if they can overcome, then they release the elicitor molecule, and to inhibit that elicitor, plant also has the urgent. So, it is a kind of zigzag model. So, similarly in case of plant viruses, host RNA silencing machinery for inhibition of that thing, virus is having the suppressor gene. So, this virus has 3 suppressor gene, V2, C4 and C2. So, there are 3 suppressors gene of this virus to inhibit the silencing. So, we choose a region which overlaps between V2 and V1, so that if we target this region, we can directly targeting 2 RFs at a time. Similarly, we target the C1 and C4, C1 is the replicase protein and C4 is again a suppressor protein. So, if we target the suppressor protein, it will weaken the virus to resist against the silencing and as well as replication and coat protein. So, all these three important regions first we choose, that will develop the whatever spacer sequence from three important region. Then manually we choose the GG sequence, the NGG sequence from this intergenic region, and these are the different spacer sequence which we took initially. Then from V2 V1 wall overlap, so this is the region we choose this many spacer sequence, and this is the C1 C4 overlap where we choose this many sequence. So, these are the initially by manually we choose, these are the could be a putative spacer sequence. Now, whether they are really suitable for doing genome editing, we do a blast analysis using the Nicotiana benthamiana as a genome to see that whether there is any match of those spacer sequence in the Nicotiana benthamiana genome. So, these are the spacer sequence, the PAM sequence, target match in the genome. None of this sequence was not directly targeted, directly available in the genome, but definitely many of them has seed sequence match. Seed sequence means the first 12 nucleotides. So, that this seed sequence match is there, and among them few are also matching with the PAM. So, these are the sequence. If you will take those sequence, then they will definitely cause a kind of off target effect, but other things, as there is no PAM match, we can choose those sequence for as a putative PAM putative spacer. But after that also second level of restriction was GC percentage. So, we choose those spacer which is having relatively high GC percentage than the others which is having less GC percentage, because ultimately it has to bind with the RNA has to bind with the DNA. If the GC content is less, then the TM will be less, the binding will be less. So, we choose the high GC content. So, in that way finally we choose these 6 spacer sequence, 2 each from these 3 regions. So, spacer 1 and 2 from IR region, 3 and 4 from this region, 5 and 6 from this region. So, this from these 3 regions we choose 2 to each 6 spacer sequence. So, after choosing the spacer sequence, next thing is that whether all these 6 spacer sequence are effectively able to cut the viral DNA under in vitro condition. So, we did a in vitro efficacy analysis, as I have told in your practical, that how we do. So, we designed a primer first in the viral genome, so that we can amplify a target region. Okay. So, which this target region will have all these spacers target sequence. So, this is the 1.5 kb sequence we have amplified like that. So, these are the region in the viral genome. This is the IR region, because this IR is having 2 part, one is this part, one is this part. Okay. So, this is YOLO is the our origin of replication region, and this red one is the spacer sequence. This is the V2 V1 overlap, and this red one are the spacer sequence, and this third one is this gray color, and these 2 are the spacer sequence. And this is the primer which we have designed, so that it can amplify the this portion, not this portion. So, this is the portion which will amplify, which is 1.5 kb size. Then the 20 nucleotide spacer sequence which we have chosen, which have identified, it was synthesized as CRISPR RNA through integrated DNA technology service, and universal tracer RNA and Cas9 protein also purchased. So, CRISPR RNA and tracer RNA were joined together to form gRNA. We have done this practical. Then this was added with Cas9, which will produce a RNP complex, and then we add the target DNA, which will cut our target DNA into 2 to see that whether it is cutting. So, this is the genome, and this is the guide RNA target. This is the 1.5 kb amplicon. So, when we give this individual 6 gRNA very nicely as per their size, they give cut, and this is the uncut molecule. So, individual gRNA can effectively cut the DNA, viral DNA. Now, the question is, our target is to do multiplexing. So, if we do put multiplexing condition, then also what will happen? So, we took only duplex DNA and triplex DNA, not more than that, because the combination will be much more. Even in duplex DNA, we have 9 combination of duplex DNA with the 6 RNA, 6 gRNA, and 2 triplex combination we have chosen, and you can see so many cutting. So, if you use 2 at a time, so viral will be chewed up by so many cuts. So, now you can imagine if viral will inside the plant, viral genome will cut in so many fragments, is it possible to religate all those molecules exactly to form a new virus? So, that was our hypothesis, that if it will cut in so many fragments, definitely there is very least possibility that viral molecule will again regain as a functional molecule. So, all this gRNA could clip the viral DNA individually or duplex and triplex condition. When we find that, we thought that we will go with all these 6 spacer sequence to develop the in planta expressing construct. So, development of the multiplex CRISPR-Cas9 construct in binary vector for localized transient assay. For that, as I have shown you this also, this paper we have followed, 37 vectors are available. We took those vectors from AdGene and developed what I have shown you in the practical. That first step is that in those spacer sequence we add few nucleotide extra to make it compatible with the golden gate system. Okay, we add those things were given in the paper, that if it is U6 promoter, what you have to add, if it is U3 promoter, what you have to add. So, like that we have added those things and prepare for every spacer sequence. We prepare 2, one is called top, one is bottom. Top means the original spacer sequence, another one is the complementary sequence of that, and then this molecule was phosphorylated at the 5' end through polynucleotide kinase, and top and bottom oligos were annealed to form a duplex molecule of the spacer, so that we can clone that. Then those molecules were cloned in this 3 vector system, 131, 132, 133. The spacer 1, 3 and 5, that means one from here and one from here, were cloned in this vector under U3 promoter. Then in the second vector we clone the other 3, and in the third vector we clone only the spacer 5, so that we can use it for triplex making. These 2 for the duplex making, this one is for the triplex making. So, all this were confirmed by colony PCR, and finally we confirmed it by the sequencing. So, the construct which gave the perfect sequence we choose for the next step, that is the multiplexing of this gRNA construct through golden gate technology. So, the uniplex gRNA, individual gRNA were mixed together in this second vector, which is called 142 or 143. If it is duplex, then we use 142. If it is triplex, we use 143 series of vector, and then we ultimately develop 9 duplex molecule with different combinations of duplex and 2 triplex molecule. So, these 9 duplex and 2 triplex construct were developed as a multiplex construct, and they were confirmed through restriction digestion. And finally, the third step, we have to join this multiplex gRNA with Cas9 molecule and transfer them into a binary vector using the gateway technology. So, this Cas9 molecule was joined using different combination of this gateway system. The multiplex and the multiplex gRNA and the Cas9 molecule was transferred in the PR ligand vector under this 35S promoter. So, this is our actually plant inoculable construct. So, this was also confirmed. So, we did the agro transformation of this 9 duplex gRNA with Cas9 and 2 triplex gRNA with Cas9. So, we agro transform them, make it ready for inoculation. So, next step is in-plant activity analysis of this construct to inhibit the chili leaf curl virus. So, this 9 duplex gRNA with Cas9 and 2 triplex gRNA with Cas9 was inoculated along with chili leaf curl infectious construct, but we did it in a spatially different way, because this is in the DNA construct. So, if we directly mix them together and add, so at the plasmid level it will cut. So, we will not able to know that whether in the replicating virus it is giving cut or not. So, what we did, we inoculate the gRNA in the bottom portion of the leaf, and we inoculate the virus at the top portion of the leaf. So, virus will come out from the plasmid backbone, replicate, and when it will started moving and entering this region, the gRNA is ready for that, and all the analysis we then did from this region. So, in today's practical you will do this agroinfiltration of this gRNA and Cas9, gRNA and virus infection in two different location of the leaf. Individually I will try to give you one thing so that you can infiltrate in the plant to see that how the infiltration takes place. And then all the analysis we did from this gRNA inoculated region. So, we did the qPCR and qRT-PCR assay to know that whether the inhibition of the virus is taking place, whether Cas9 and gRNA is expressed properly. So, what we observed in case of virus accumulation, the chili leaf curl virus qPCR, this is only the chili leaf curl virus infected sample, and these are the gRNA treated chili leaf curl infected sample. In all these cases we observed significantly the virus accumulation is reduced, and in 3 cases at least, in these 3 cases it is reduced very significantly. So, that but the question is that why there is difference, whether there is difference in the Cas9 and gRNA expression. To do that, we also analyze the gRNA expression and Cas9 expression in all these plants. We observed there is no significant difference in the gRNA and Cas9 expression in this tissue. So, in all the plants they are almost equal, because this is only transient expression, not transgenic. So, definitely this much variation we expect, because in all the leaves it will not definitely express similarly, because it is not a transgenic condition, but they are not having any significant difference. So, whatever cutting is taking place, they are due to their ability to cut inside the plant, how they are cutting. Now, the question is that if there is so much reduction of the viral load in the plant, whether there is any impact in the symptom. We do not expect, because we are only doing a transient expression, only in one leaf we are inoculating, it is not throughout the plant. So, whatever virus is present, it is not expected that some of the virus cannot escape and cannot go, wild virus, not the mutated virus. Some virus will definitely escape that only that zone, and then they can move throughout the plant, because in the other leaf of the plant there is no CRISPR-Cas9. So, in other leaf of the plant it can happily replicate, happily produce the symptom. Still we try to see that as there is very huge reduction of the virus load, will that be sufficient that the virus will not able to move throughout the plant and cause the disease. So, we try to see what is the impact in the symptom. So, symptom analysis has been done. We observed that the virus is causing symptom in curling of the leaves and stunting of the plant. 2 types of symptom actually you observe. This is the healthy plant. So, this is the condition that a curling of the leaf and very much stunting of the plant. So, these are the 2 symptoms. So, we divided this symptom in 3 grades. What are these grades? In the leaf curl, if there is no visible symptom, we give the grade 0, then flaking, vein thickening and vein twisting 1, mild puckering and folding of leaves from margin 2, and cupping, rolling of size reduction of the leaf, it is 3. Similarly, in stunting, if there is no stunting 0, stunting less than 20% 1, stunting 20 to 50% 2, and stunting more than 50% 3. So, these are the symptom category. We have devised that thing to know that after inoculation whether any of this category is showing any improvement, because if directly we inoculate the virus, the category is 3 and 3. The stunting and the curling is 3 and 3. So, whether we can improve that category to somewhat upward level. So, first thing what we have observed, that symptom appearance delayed by 15 to 20 days. Normally when you inoculate the virus, it will start producing the symptom within 7 days, but in the gRNA treated plant it is delayed by 15 to 20 days. So, it is only a transient inoculation, not even transgenic plant. So, if it is transgenic plant, definitely that will give you much more protection. Then symptoms are less severe in all gRNA treated condition, and 3 combination of gRNAs where viral accumulation drastically reduced also exhibited almost no symptoms. So, these are the 3. So, among this you can see this leaf curl 0, stunting 0 in gRNA 5 plus 2. 5 is from the C1 C4 overlap and 2 is from and the V1, V2 overlap. So, that big chunk is deleted out. So, it is very difficult to virus then cope up, and then followed by this 5 plus 4 and then 1 plus 2. This is the origin of replication, where leaf curl is 1, stunting is 2 grade, and here 0, 0, 0, 1. So, you can see this is the picture of those symptoms. So, these are the, you can see this is the best combination, this is the control, an empty vector control. It is almost same as empty vector control in the height as well as the leaf curl symptom. This is the chili leaf curl virus infected plant. This is the second best combination, and this is the third best combination, which we observe that in this combination the virus load also decreased severely, and the symptom is also not there. So, this is much more clear view that there is no leaf curl symptom also in this 3 combination. And the question is that, do virus accumulation also reduce in systemic leaf? So, we have checked only in the inoculated leaf the virus accumulation reduced. So, whether it is reduced in that systemic leaf also, that virus cannot able to replicate in the systemic leaf also. For that we did 2 experiments, rolling circle amplification of the virus, so that to know that whether virus is able to replicate in this leaf, systemic leaf. We observed this is the control, only the virus inoculated sample, there this much concentration of the virus accumulated, here very less amount of virus is accumulated, which is further substantiated by the qPCR assay, where we showed that this plant it is almost negligible, and then second plant is this and third plant is this. So, compared to the only chili leaf curl virus infected condition, the virus accumulation even in the systemic leaf, though we have not treated in the systemic leaf, only we have given the injection in the one leaf, but it is showing its effect even in the systemic leaf. So, as this condition is there, so definitely we have this question, is NHEJ generated escape mutant formation is reduced, because people have shown that due to this escape mutant formation virus can move, virus can produce symptom. So, as we are seeing the symptom is not there. So, definitely the question is there whether really escape mutant formation reduced or not, and if escape mutant formation reduced, that means we will not able to see any genome edited mutant virus. What is actually the aim for chromosome or host editing? We target for that to see that whether editing is there, whether any mutation is created in those position. But in case of viruses, we do not want to see any mutated virus, we do not want to see any sequence where mutation is there. Okay? So, it is a little bit different from the host genome editing, because there we want to see the editing is taking place in the chromosome, but here if editing is there and if mutant is there, that means virus can withstand that editing. So, we do not want to see any edited virus. So, the technology is available for that. For that we have to do the assay for the genome editing, that we have done through T7E1 assay and sequencing. The T7E1 assay will be discussed by Dr. Vishwanathan in practical. So, what is the basic concept is that if you have a original wild virus or a wild sequence and a mutated sequence where a mutation is there, then you just mix them together and heat denature and slowly cool down. So, some of this molecule will form heteroduplex with respect to only few nucleotides. And there is one enzyme called T7 endonuclease 1, they can detect this kind of heteroduplex, and if there is heteroduplex, they can cut from that heteroduplex region and they can generate this kind of fragments. So, what we expect that if that is the band, then PCR band, because in the infected plant, for example, in the infected plant there will be some virus which is edited and rejoined, and there will be some virus molecule which is wild. So, when you do the PCR amplification from after the treatment, when you do the PCR amplification from those inoculated region, you will get the amplified product, both the wild virus sequence as well as the mutated virus sequence. But we do not want to see any mutated virus sequence. If mutated virus sequence is there, that means the editing cannot cause any inhibition effect to the virus. So, but in case of plant, if you want to do host editing, you want to see this kind of band. So, this is one and this are the two bands. So, that means editing has taken place properly, but in our case our main aim is to, it should only reduce the virus load, it should not produce any mutant virus. So, when we did this assay, this is the control which is showing this band, but in this plant we only see only one band, there is no mutant molecule generated. To confirm these things, again we took this the same PCR product for sequencing. Now, as Dr. Reddy yesterday told, that after sequencing, if there is a mutation, you will get two types of peaks. So, what we did, we took the viral genomic portion, amplified and sequenced, and sequence of wild virus and mutated virus, if escape mutant forms should be presented, should be there if there is a mutant virus. So, there is a software called TIDE. Using this software, you just have to put your both the sequence of your wild virus as well as mutate means your spacer sequence and the sequence of your only the virus inoculated plant and the gRNA treated virus inoculated plant. From both the things, the PCR amplified product you have to sequence, and you have to load this sequence here. If this is the control sequence, and if there is a mutant infected pool, then you will get this kind of different mixed kind of peaks after the PAM motif. If there is a, if the virus is joined together and again replicating, then you should expect this kind of peaks. So, after analysis, what we observed, that this is the PAM motif and there is very few negligible amount of different nucleotides are present, which is not at all significant, because if it has to be there, it will be big peaks in here. So, as the big peaks are not there, so this indicate that there is perfectly no escape mutant formed after the multiplex editing. So, conclusion, multiplexing of the gRNAs were attempted to inhibit chili leaf curl virus infection in Nicotiana benthamiana. Using the golden gate and gateway cloning, 9 duplex and 3 triplex gRNA combinations were developed with Cas9 from 6 spacer sequence identified covering origin of replication and other important genes of the virus. Among the 11 combinations, 3 found effective to reduce symptoms in the virus accumulated plant, and remember that the triplex is not giving that much valuable more reduction. So, it is not that if you introduce more gRNA you will get a very good result. So, in general all the gRNA inoculated plant, the viral titer was lowered as observed in qPCR analysis, and T7E1 assay and sequencing revealed the reduction of escape mutant formation, and that may be due to that virus after cutting they may not able to religate and form a significant virus molecule. So, this is the summarized result of what we have done, and I am thankful to Dr. Hanu Papu of Washington State University, who provided all this facility to conduct this experiment, and this is Dr. Neff. Actually his lab is working on genome editing, Dr. Papu's lab is working on the virus. So, I took all this vector from this lady who is a PhD student who provided me all these vectors, and I worked on his lab to conduct this experiment. Thank you. Any question? Yes, because I have heard that someone of you will be doing virus editing also. So, you can see those papers and you can.
So, thank you for your lecture, sir. Sir, my question was like this intergenic region, it is said to be conserved across the species or the genus of begovirus.
There is one non-nucleotide sequence, 9-mer sequence, which is conserved across all the single stranded DNA virus.
So, if
in the Gemini viridae family, they are almost conserved.
So
but the IR region is around 200 nucleotides. So, that is not conserved, but that non-nucleotide sequence is conserved, but that is conserved within a species, that is conserved definitely, but one species to another species is different.
So, if
chili
we
leaf
design
curl
a guide
virus
RNA,
IR, chili leaf curl virus IR and tomato leaf curl virus IR will be different, but within chili leaf curl virus different isolates, they are almost having the similar IR region.
So if we design this guide RNA based on this 9 nucleotides, can we target all the chili leaf curl virus?
Definitely. One of my special sequence is from there, and that is the beauty that in almost all the begomoviruses, after that mononucleotide sequence, there is a CGG. So definitely any Gemini virus you can use that mononucleotide sequence as a target, and then there will be a definitely PAM motif. So that is there. And whatever special sequence we have designed, initially actually we have aligned with different chili leaf curl virus, not only chili leaf curl virus, the other viruses which infect chili. So almost we choose those region which is conserved along all those, so that whatever construct we have developed, they should target the other viruses also. It is not exclusively on the chili leaf curl, it should be a broad spectrum. So whenever you try to develop, you first do a multiple alignment, try to find out a conserved region from where you can design the spacer sequence.
And sir, if I design a like guide RNA against two different regions of a same ORF, like suppose
Yes.
code protein region, will that be more useful or the multiplexing will be more useful?
No, I have whatever things I have designed two things from the same region. Okay. For example, V2 V1 overlap. From that I designed two. Then code protein, replicase protein and C4, that region I developed two. But what we observed that those two spacer from the similar region doesn't give very much advantage. But if one is from this region, another from this region, then a big chunk of the DNA may come out, and that may inhibit the virus, because whatever gRNA is designed from one particular locus, two gRNA may be, but we observe that they are not that much effective. Definitely all are effective, but if you compare, then the gRNA from one side and another side is much more effective than from the similar side. But all these things we have done in a transient condition. If you do it in a transgenic mode, they may be completely different.
So sir, this case where we target the virus is always transgenic only?
Yes, because when the virus will come, then only already the plant should produce those CRISPR and Cas9, because it is not a plant genome that you can develop and then segregate the things and develop a edited plant, because that can only be possible if you target the host protein like EIF4E. In that case that is possible, but if you want to target the viral genome directly, only the transgenic is the means.
Thank you, sir.
Any question?
Thank you so much, sir, for your lectures. By seeing your lecture, we can target plant like by viral infection. We can target plant at any stage of plant. So my question is that, sir, through viral infection, if we are targeting a particular viral disease, so when virus is targeted and that viral genome is edited, and the plant will not produce the symptoms of the viral infection. So how it will be transferred to their generation to the next generation of the plant? Will it
if you develop a transgenic plant, then definitely it will be. If it is only transient, means you cannot do.
No, means if that viral genome is targeted.
Viral genome, for that you have to integrate the Cas9 and the target gRNA of the virus into the plant. Okay. Then only it will always produce those gRNA and Cas9 protein in the plant, even in the next generation also.
Next generation also.
And then also when the virus will come, then you can target that.
Okay. And my another question is that there is the vectors, they are transferring plant viruses from one plant, infected plant, to the other plant. So can we use that viral vector for transferring our construct also to one plant to other plant? Like this is
actually this viruses are transmitted by the vector through some specific interaction of the gut protein of the insect with some of the viral coat protein. So without that interaction, nothing can be carried. So can you imagine, you just go to any field, the mosquitoes are so much in the field, but none of the mosquitoes has been reported to transmit any virus, because there is any plant virus. So, there is a very much specificity of the insect with the virus. So, the white fly transmit the bigoma viruses only. Okay. So, if you try to infect the white fly with your body, it cannot carry the dengue virus to transmit. So, similarly, so if you want to transmit your CRISPR-Cas9 reagent, that cannot be recognized by the white fly, and that cannot be transmitted.
Okay.
Okay,
Thank
if
you,
that is so easy, then people would have used that kind of things.
Yes, sir. Okay, thank you, sir.
Any question? Yes.
Sir, my question is, if you are using virus based delivery system, whether it can be systemic to the next generation also, because
The question is that whatever DNA virus we are using, definitely that cannot be transmitted. But there is one RNA virus called TRV, which because people are using that thing, though we are not able to use that because that virus is not present in India. So we cannot use those things due to quarantine issue. So, but in TRV, that virus can enter even in the seed. So, if any virus which can infect the seed, then it can be transmitted to the next generation. But if the virus is not going to the seed, for example, this bigoma virus or CGMMB, what we are doing, those CGMMB we are seeing the virus is present in the seed attachment site very much. The concentration of virus is very high. So, we do not know whether it will carry in the next generation. That is after only experiment we can know, but definitely TRV is going, and other viruses, there are several other viruses which is seed transmitted virus. So, in those cases, if you develop a vector, they definitely go. But in case of plant viruses, the major problem still now, we cannot deliver Cas9 till now using the virus based vector, because every virus has a limited cargo capacity. So, above that they cannot carry any gene and express, they will throw out that gene. So, only we can deliver the CRISPR means gRNA construct, but the Cas9 has to be expressed from the plant. Till now there may be possibility that in future we may have a second version of Cas protein which is smaller, because already in Staphylococcus aureus the Cas9 protein is very small, not like this much big as Cas9. Okay, so and they are having two different polypeptides, so you can even put two polynucleotides. One is 1.3 kb size, another one is 1.5 kb size, like that. So exactly I am not remembering. So very easily that much DNA you can clone it in a virus based vector, but not the 4.5 kb or 5 kb. The size of the Cas9 is, if you add the NLS and the 35S promoter and other things, the size is around 5.5, like that. So it is very big. So that much DNA we cannot clone. We can clone, but it cannot sustain inside the plant. The virus will throw out. That much big.
thank you, sir.
Any question more? So who are directly will be working in the viruses, for them I am telling you that you can consult me in future, that I can help you how to design, how to do the editing construct to show the things.
So, any more questions? If not, let us thank Dr. Anirban Roy for his excellent lecture with this momentous.
For me also,
Yes, sir. Thank you, sir.
yes. Photo.
After this, we are having feedback, and from after feedback, we are going to go to genomics.